All right, today we're watching a fascinating showdown. In one corner, you've got a classic data crunching champion, and in the other, a brand new deep learning contender. And their battleground? Well, it's the microscopic chemical universe that's actually inside our own bodies. So let's dive right in. Think about it. Every single cell, every bit of tissue in our bodies is just humming with biochemical activity. It's like a story being written in molecules, but the whole story is invisible to us. So how on earth do we read it? How can we possibly turn this invisible world into a map, something that could let us spot a disease before it even really gets started? And that is the core challenge researchers are up against. They're trying to create a super detailed chemical map of our tissues to figure out what's going on with our health. Sounds simple enough, right? But the complexity is just staggering. Okay, take a look at this right here. This is a little slice of human colon tissue. Visually, yeah, you can see some different structures. But chemically, oh man, this tiny little piece contains an entire universe of information about our metabolism. The real key, though, is figuring out how to unlock it. So, to do that, scientists use this really powerful technique called mass spectrometry imaging, or MSI for short. The easiest way to think about it is like, it's a chemical camera. It scans a tissue sample, and for every single tiny pixel, it records this incredibly detailed profile of all the molecules that are right there. And when I say detailed, I am not kidding. For every single pixel on that tissue map, the MSI generates over 20,000 different data points. That is just an unbelievable amount of information to deal with. So, in the world of data science, this has a name. It's called a high dimensionality problem. And really, it's just a classic case of data overload. You have so much information that the important patterns, the stuff you're actually looking for, gets buried. And what makes it even harder is that the relationships between all these molecules, they're not simple, straight lines. They're these complex, tangled webs, what scientists call non-linear relationships. Trying to find a disease signal and all that, it's like trying to find one specific knot in a giant, messy ball of yarn. So you might be asking, how have scientists traditionally handled this? Well, for a long time, they've relied on this trusted, old-school workhorse of an algorithm. So let's meet the reigning champion, PCA. PCA, which stands for Principal Component Analysis, is a statistical method that's been the go-to tool for, decades. Its main job is to take all that crazy high-dimensional data and just simplify it, kind of boil it down to its most important parts, so we can actually see what's happening. But, and this is a big but, it has a crucial weakness. And here's the thing. It's all about how PCA simplifies the data. See, it only looks for linear patterns, these simple, straight-line relationships. But remember that tangled ball of yarn? PCA is trying to understand the whole thing by only looking for the straight threads. It just doesn't work. A really great way to think about it is this. It's like trying to understand a complex 3D sculpture by only looking at its flat 2D shadow. Sure, you get the basic shape, but you lose all the depth, the curves, the texture, all those non-linear details that are actually the most important part. And this picture right here perfectly shows the problem. When you run PCA on this tissue data, this is the result. I mean, look at it. The image is noisy, the different parts of the tissue are all blurry, and the really important structures, they're almost impossible to make out. It's just not good enough. Okay, but this is where our new contender steps into the ring. It's a much smarter, more sophisticated deep learning approach. It's called a variational autoencoder, or VAE. All right, I know this diagram might look a little complicated, but the idea behind it is actually pretty elegant. Remember how PCA only sees the flat shadow? Well, the VAE is designed to understand the whole complex 3D sculpture, the real nonlinear messiness of the data. So let's break down how it actually pulls this off. It basically works in 3 steps. First, you have something called an encoder. It takes all that massive 20,000 point data for each pixel and is forced to squeeze it down, compress it into a tiny, efficient summary. This little summary lives in what's called the latent space. Then a decoder has to take that tiny summary and try to rebuild the original complex data perfectly. And that is the really crucial part. See, to get good at this game, squeezing the data down and building it back up, the VAE can't just look for simple straight lines. It's forced to learn the real underlying rules of the data, all the complex curves and interactions, those non-linear patterns. It's kind of like an expert artist who doesn't just copy a face. Instead, they learn the deep rules of facial structure, which lets them draw that face from any angle. The VAE learns the sculpture, not just the shadow.
All right, so we've got our classic tool, and we've got our new contender. The researchers decided to put them head-to-head on the exact same set of human colon tissue data. It is time for the main event. So in one corner, you've got PCA, the linear method that's just finding the shadows in the data. And in the other corner, you have the VAE, the nonlinear, deep learning model that actually learns the underlying structure. And the results are just wow. Seriously, look at this side-by-side comparison. On the left, that's PCA's chemical map. It's a muddled, noisy mess, right? And on the right, that's the VAE's map of the exact same tissue. The difference is just night and day. The structures are sharp, they're distinct, they're vibrant. It's incredible.
Okay, so the next test was something called clustering. That's basically grouping similar pixels together to find distinct types of tissue, which, could be the difference between a healthy area and a diseased one. And here's PCA's attempt. The boundaries are all messy, and it just completely fails to identify key structures that we know are in that tissue sample. But now, look at the VAE's clustering. It is incredibly precise. The clusters that it finds line up almost perfectly with the actual physical structure of the tissue. It's finding real, distinct biological regions that PCA was totally blind to. So the visual evidence is pretty compelling, right? But the hard data really seals the deal. This chart shows what's called reconstruction error, basically how well each method could rebuild the original data after it squished it down. And for this, lower is better. As you can clearly see, the VAE's error is just consistently and dramatically lower. It just does a way better job of understanding and holding on to the really important information.
So yeah, the VAE is the clear winner here, no contest. But what does this victory actually mean for science, for medicine? Well, it means we're getting a powerful new lens to finally see that invisible world inside all of us.
So let's just recap the VAE advantage real quick. It actually captures those complex, nonlinear patterns that are at the heart of how biology really works. It compresses and rebuilds data way more faithfully. And because of that, it allows for this super precise identification of different tissue subtypes, and it reveals these hidden chemical signatures that older methods like PCA were completely blind to.
So why does any of this matter to you and me? Well, because these hidden structures that the VAE can see, those are the keys to understanding disease. By creating these much clearer, more accurate chemical maps, this tool can help scientists discover new metabolic pathways, pinpoint disease markers, and ultimately develop faster, more precise medical diagnostics for all of us. And that brings us to a final, pretty thrilling thought. We've just witnessed a major leap forward in our ability to read the chemical stories written inside our own cells. So if this is what we can see now, just imagine what other profound biological secrets we're on the verge of finally unlocking.